What I'd like to do is first welcome everybody to what is essentially our 4th seminar in our UCD COVID-19 seminar series. The purpose of this series is for that we've built upon from the UC Research Partners team is to try and aim to showcase some of the great COVID-19 related research that takes place at UCD across all of the different colleges in the university. But also it's a means to hopefully encourage further interdisciplinary collaboration between UCD and external partners and within UCD, but of all our internal academic partners as well. During today's seminar, the kind of format is going to be, we're going to hear from our speaker after I introduce her, and then we'll conclude with a short Q&A session at the end. So please submit any questions that you may have along the way via the Q&A function in your browser. Okay, so today our guest speaker is Dr. Virginie Gautier, and she is an associate professor in virology, and she's also a principal investigator in the UCD Center for Experimental Pathogen Host Research, which is directed by Professor Paddy Mallon in the School of Medicine in UCD. Dr. Gautier leads a research program focusing on HIV molecular pathogenesis, the HIV host virus interface, which is funded by the HRB, IRC, and Enterprise Ireland. And she's also the coordinator of the Horizon 2020 EU for HIV Cure research program that looks to accelerate the HIV cure research in Europe. Now, following the COVID-19 pandemic, Dr. Gautier shifted her research focus and developed a dedicated COVID-19 program with collaborators within UCD School of Medicine, Veterinary Medicine, Chemistry, and the School of Molecular and Biomedical Science, and also in engineering. So critical research actions that include the SARS-CoV-2 molecular diagnostics, antiviral drug screening, neutralizing antibodies characterization, and PPE surface validation, which has all been made possible due to the Irish government's COVID-19 rapid response scheme. So at this point, I'd like to pass over to Virginie. Today she's going to be talking about her research that focuses on her last 9 months of this year, which is focused on the mobilization and the engagement at UCD Center for Experimental Pathogen Host Research that addresses the COVID-19 pandemic specifically. So Virginie, thank you very much for joining, and I'd like to pass over to you to talk a little bit about your work.
Thank you very much, Kevin, and thank you for the invitation to share all this knowledge with you that we have accumulated over the last 9 months. So let me share this with you. Whoop. So as Kevin mentioned, my focus of research previously was more on HIV and HIV cure. So it's a virus, but it's certainly not a coronavirus. And so when at very earlier stage of the pandemic, it became quite evident that we were in a unique position to actually respond to the pandemics and actually devise some research strategy and action just to help. So here I just want to quickly acknowledge CEPHR, so the Center for Experimental Pathogen Host Research. And one of the key reasons why we were able to respond so quickly is because we host the Containment Level 3 lab, which enable us to work with pathogen like SARS-CoV-2 or HIV and so on. So from a research point of view, I think that was a key determinant. Another one is like because we are involved in HIV research and clinical studies, and this is more the area of Paddy Mallon, the director of CEPHR. We already had in place all this clinical expertise and clinical studies. And certainly the biobanking facility here to actually collect all the patient samples, like COVID-19 patient samples. And that's the core, because then you have access to all this crucial material to ask different research questions, whether it's immune system or virology-based questions. So in UCD first, when it just first arrived, because it was kind of an experience in itself, is that we realized that the first thing is that, of course, big, huge global health problem associated with infectious disease. So directly associated with us, and it was very urgent to rapidly respond. And the first thing that was very important was to make sure that we could sustain a good diagnostics facility. And we really realized shortly that we had an issue of reagent availability and shortage given the high demand across the world. So it was happening in Europe, in the States, and of course in Ireland as well. So the UCD COVID-19 task force was quite helpful in the sense that we could try to coordinate and identify the need and then coordinate the response. And if it was as simple as making up a buffer and delivering it to the hospital so they could just carry on with the COVID-19 diagnostics. And then it just became obvious that it could be more serious shortage of full kits maybe and so on. So that's where we decided to develop collaboration to actually develop kits ourselves just to make sure that we wouldn't face the same situation again. And then another aspect as well that is quite important are the PP or surface development in terms of protection and COVID-19. Another one, which is our specialty, is also the host pathogen interaction and how you can capitalize on your knowledge of how the virus infects the cells to develop antivirals. The vaccine, we are actually not involved. That's not necessarily our specialty. But then the other thing that we were quite interested in is to develop novel antivirals. So we've been doing this with HIV. So all we had to do is just like shift the focus and translate what we knew already about how to develop cell-based assay to develop then antivirals. And we are also involved in neutralizing antibodies and not so much into the development of anti-inflammatory medication. That's more something that is towards the hospital. And it became also very evident that it was important to be maybe better prepared and ready for the next pandemic when it will come to us. So I'm just going to give you like a short overview of what we've been doing and working on so far. And mainly two major projects that were funded by the COVID-19 response, thanks to the government. So this one was funded by SFI, and it's a collaboration between Gil Lee and Paddy and myself. So the advantage here was really to have access to the expertise of Gil in the School of Chemistry, who is especially in nanotechnologies, and had developed these excellent magnetic beads, which are coated with silica. And these are excellent to actually capture the RNA, whether it's from any origin, actually, nucleic acid like DNA and RNA. So here the idea was really to sustain the potential need and address the potential shortage of kits and reagents for viral RNA extraction, which is essential for molecular diagnostics. So the first remit was really to develop a SARS-CoV-2 isolation workflow. What we had to work and optimize at the beginning was a lysis buffer. So the idea of the lysis buffer is first to inactivate the virus, and second to release the RNA from which is encapsulated into the viral particles, and also to inactivate RNases, which are going to eat away your piece of RNA. The second part is actually to extract and to capture the RNA from this lysate. And to do this, you can do this, as I mentioned, with these magnetic beads coated with silica. And then once they capture, you use the magnetic capacity of the beads to actually wash the beads with different washes buffer to remove everything you don't need, like protein and lipids and so on. And then finally, you elute it. The second remit was to actually develop a multiplex RT-qPCR, which was targeted specifically to the SARS-CoV-2 genome, targeting it at two different places, and a third combination of primer probe as well to look at a human gene, just to control for the RNA extraction procedure. So then we quickly tested it against what is well known, the QIAGEN kits, which is a viral RNA QIAGEN extraction kit, which is like a standard in the lab. So we had it here, so it was quite easy. And then we were very happy to see that using serial dilution of the virus we had obtained from the EVAG program, which was serially diluted, that we had a nice robust assay with a very good reproducibility, a nice specificity, and a good linear dynamic range, and a very good sensitivity as well, with a nice limit of detection. So when it's working nice in vitro, you have to make sure it's working with patient samples as well. So there it was very important to work with Paddy Malone on this one, because it has this established the All Ireland Infectious Disease cohort. And through that, we were able to have access to patient samples and just to look at how well we were able to quantify and to analyze the SARS-CoV-2 genome. So to do that, we actually work with the St. Vincent's University Hospital, who hosts the Roche MagnaPure 96 system, which is a standard in the field of viral RNA diagnostics. And it's an automated system that can process a lot of samples at once versus our manual system and workflow, and it actually performed quite well up to CTs of 35, but that's kind of a standard. Like after 35, the CTs vary a lot, and the detection is not that, so the system is usually not that sensitive. And then what was important, like a manual workflow is quite easy to install in different places, but what would be nice as well is to make sure that our workflow could be transferred to an automated workflow. So we work with the National Virus Reference Lab, and they have this Kingfisher system that works also with 96 well plates. And again, we could see that whether it was manual or using an automated system, it was actually working quite well. So we were quite happy. So now we're at the stage where we can provide up to 40,000 tests, thanks to the teams of Gil Lee in the School of Chemistry, and they have produced a large amount of beads. And if necessary, now we can provide it to the hospital in case they have any type of shortage. So here, like again, it's important to highlight the collaboration between two different fields, one in chemistry and the other one in medicine. And this one, so like just to move on another side, so we have this expertise in screening antiviral compounds as well. So we decided to go forward with that, and we received funding from the HRB through again the COVID-19 response. So when you develop an antiviral, it usually takes about 10 years to actually get a good lead compounds. And you actually go through a lot of compounds, only a limited number of them are going to pass all the stage of the testing. And it's actually quite costly. And it's so to shorten and to facilitate and accelerate the identification of potent antiviral compounds against SARS-CoV-2, you have two strategies basically. So one is to use broad spectrum antiviral agents. These are antiviral compounds that are known to be active against several viruses, and it's kind of the golden pill that you're looking for, a little bit like an antibiotics that is broad spectrum. So that's what you're looking for. We don't have any at the moment. All the antivirals really target the specificity of each different viruses. But I'm sure you've heard about remdesivir, and Gilead's have been trying to develop these broad spectrum antivirals, and that was the first one selected. And this had been tested against Ebola, for example, and it was going well up to in vitro trials, but failed in the phase 3 clinical trial. So that was one of them that was selected for first clinical trial, again, SARS-CoV-2 and COVID-19. And you had this huge clinical trial organized, like international clinical trial, the Discovery clinical trial, the Solidarity from the WHO, and then Recovery in the UK. And they all selected these like 3 compounds. So remdesivir, as I mentioned, hydroxychloroquine would be also a broad spectrum antiviral agents that was shown to be active against quite a number of viruses in vitro. And then you have lopinavir, which is actually well-known and used for HIV infection. And it targets specifically the proteases. So the idea behind using these broad-spectrum antiviral agents is either you target a viral enzymatic activity or structural protein of the virus that you can find in different viruses, because they all have a similar strategy when infecting the cells. And the other one would be targeting cellular pathway that many viruses might hijack to maximize their replication. And by targeting this host virus junction, you can actually block the replication. Another way to speed up the process is to look at drug repurposing or repositioning, like kind of upcycling your drugs if you want. So where you would take them from a different area of research or work, like in cancer, for example, or in diabetes area and so on. And then you know that they are safe to be used, either because they've been tested up to phase 3 clinical trial or because they're actually in use. And then you bring them to look at the impacts on the virus. So there you can have either the idea of screening very large compound library, and you go through all of them, and then you select 1 or 2 that are good at the end, and then you move on to clinical trial. So we try to actually work with all these different strategies, starting from scratch. Yeah, so we have this cell-based assay for SARS-CoV-2 infection. So what we're using are these Viro E6 cells, they're quite well known in the field, and they sustain viral replication for SARS-CoV-2, which is actually what you need. These are non-human cells, they're kidney, they are African green monkey, and they're also kidney cells. So they're a little bit far away from what you would expect to work with in terms of, respiratory problems and so on. So, but the advantages are they're very easy to grow, and they sustain good viral replication. So it actually makes them a good tool to test different viruses and drugs. And then, so what we do, we usually work in 96 well plate formats in triplicates. And then what we start by pre-incubating the cells with the antivirals to give a chance for the antivirals to actually reach the cells and penetrate the cells. Then we add the virus for 2 hours, and 2 hours only is just to synchronize the infection and limit it to just to give enough time for the virus to penetrate and enter the cells. Then we wash everything, and we monitor the infection for up to 48 hours. The way we monitor the infection is by looking at the reduction in viral infection, is to look at the supernatant, so the virus being released by the infected cells, and titrating this using RT-qPCR following RNA extraction, a little bit like what I was showing you earlier on. Or you can instead direct your attention to the infected cells and look at how many infected cells there are, using immunofluorescence targeting specific protein of the virus. So in our case, we actually work on the viral nucleoproteins because it's quite abundant and it's expressed very soon after the virus penetrates the cells. So that's just typical examples. So far we screened quite a number of drugs, and we are focused on 2 different drugs. One is considered as a broad spectrum antiviral that was shown to actually be effective against SARS, the first SARS, and other viruses like hepatitis as well virus. And this gave us an IC50 and dose dependence. So that's this one. And then we validate, so that's at the viral RNA being released by the cells, so the viral titer. And then we looked at the cells as well to look using flow cytometry to look at the number of infected cells and if we had this dose dependent effect when we add increasing amount of the antivirals. So that's your DMSO control, where you have about 40% of infection at 24 hours. And then you see that when you have very large amounts of the drugs, you limit the extraction like to 0 nearly. And then while you decrease the level of the drugs, the antivirals you use, you see that the impact on the number of viral infected cells decreases. And here is kind of the same idea, except we use fluorescent microscopy to look at the impact. So we always compare our drug to known drug like hydroxychloroquine, which work very well in vitro, remdesivir as well, which works very, very well in vitro. So like you look here, dose dependency of the activity of hydroxychloroquine. So in green, you have the infected cells. Beside it, that's the total number of cells. And you can see that as you increase the level of drugs, you increase the inhibitory capacity of the drugs, the antiviral capacity of the drugs. And here you have the same thing. So this cell-based assay can also be used to measure the capacity of antibody to neutralize the virus. So neutralization assays using similar approach, except here you would pre-incubate the virus with the antibodies or the plasma, the convalescent plasma from individual positive for SARS-CoV-2. So you would pre-incubate the virus and the neutralizing antibody, so it would block the virus, stop it from infecting the cells, and then you would monitor the impact as well later on. So that's something that is going on at the moment. So we're screening some plasma from patient samples. So that's just 2 examples, but we have many, many more actually through collaboration. So the advantage here is also to work with collaborators who would be expert in some specific cellular pathway that the virus would hijack to infect the cells. So that's where we have developed other collaboration as well. So then the next stage is to be maybe better prepared for the next one. So I think we saw the great advantage of having a biosafety level 3 facility to actually work with the virus, given it's a risk group 3 biological agent. And I have to say, I work very closely with the similar facility that is in the vet college. and we made sure that we were working together and then, at the same time. And the two of us were the two of the first CL3 laboratory ready and having all the authorization to work with SARS-CoV-2 for research. I think that was a great first step that enabled us to go a little bit further down the line. I think what would be needed, I think that would be great. The next stage would be to have an animal facility up to this standard, so you can actually work with animal models and infection and just to test drugs and to study the disease simply and how the virus takes over the whole organism. So here are all the names of the people. So from CEPHR, directed by Paddy Malone. Here are the most of the people that were involved in the UCD COVID-19 response. And that was very important at the beginning, just to help make connection with the different people. And that was organized by UCD Research and shared by David McHugh. And then that's people from the NVRL. So we have great capacity in UCD. It's unbelievable in terms of viral knowledge and expertise in viruses. So they were of great help as well to support us when we developed the diagnostic assay. The School of Chemistry, as I mentioned. So we're actually going further from the initial kits, and now we're looking at modifying the chemistry, the surface chemistry of the beads to actually be able to extract the viral RNA even more specifically and being maybe more sensitive as well, with Dermot McBroken and Suzanne Quinn in the School of Chemistry as well. I'm working closely with Camillus Power from the Tala University Hospital because he was very interested in specific drugs targeting the virus, and we thought it was quite good as well. And with Emmanuel McCoy and co, we're actually working on PPE and how it can accelerate maybe the decay of the virus in different environments. Working with peptides now and Denise Shields using them as antivirals with Marc Devaucel in RCSI. With Margaret McKee, we're going to look at specific cellular pathway that we could target to interfere with viral replication. And with Margaret Warhol as well, like we're looking at how to interfere with viral entry, for example. And with Liam Armand and Aurélie, we're looking at developing and testing a wide array of antibodies against SARS-CoV-2 as diagnostic tools with immunofluorescence and so on. So that's just want to thank all of them, and I'm sure I've missed a few of them, so apologies if I did. And thank you very much. I think we should be ready for questions.
Great, thank you very much, Virginia. It just goes to show the scope of the various different work going on that you're involved in, and that's not to say there's others as well who are involved in so much work around COVID-19. The sheer number of names, and to think you may have missed out on a name or two is amazing. So well done on everything you've done to date, and well done to all your collaborators. I might start off with a question in relation to the antiviral work. I'm just wondering, you're looking there at various different in vitro sort of studies, and I was just wondering around the difficulties that may be in place in terms of transitioning from that sort of research into seeing efficacy in clinical trials.
Yeah, so that's actually a very good question and a tricky one. So how do you know, like from the in vitro work you do? So if you see, like I think it was the case with remdesivir and hydroxychloroquine. So in vitro, they were actually giving excellent results. And we tested them ourselves, and we find our drugs actually matching or sometimes bettering the capacity to inhibit viral replication in these cellular models. So I think what is important is to understand that there is a huge gap between that's why I didn't put the names of the drugs, because I don't want people to get their hopes up. The idea is that, yeah, then it's a huge gap. So the next stage would be maybe to go through better cellular models. And then, because this one is good for high throughput screening, but then you can go a little bit better in terms of human cellular models and primary T cells. And then obviously testing it in, the first step will be to test it in animal models. And what is interesting is that remdesivir had been tested against the first SARS in animal models when it was selected initially early back in January, February by the large international clinical trial. So that was a good positive point for remdesivir. Hydroxychloroquine hadn't been tested in animal models and was subsequently, and it was shown that using macaque, so non-human primates, it's actually not effective, and it's not working that well in primary human respiratory cells as well. So I think it's important to test all of them before you can move to the clinic. Earlier on in the year, I think we couldn't really wait to do all these tests to move on to clinical trial. And because these drugs were safe to use in human, I think it was important to move forward quickly.
Mm-hmm. And is there a risk then as well in terms of the potential for SARS-CoV-2 to mutate in terms of resistance to any of the antivirals that you're kind of focusing in on?
So that's another very good question. So these viruses are RNA viruses, and they have this capacity to mutate very quickly and to adapt to the environment. So if in the environment or in the cells you add this drug that is inhibiting it, they might find a way to overcome this inhibition and to find a different route, basically. So what you can do against that, and that's what, I didn't show the data here, but that's what we've been doing as well, is that you don't just use one drug, you actually use two different ones, two different classes of drugs targeting the virus at two different sites. And then the chances that the virus will be able to evolve to counteract these two mechanism at once is very small, and then you can even use 3 drugs if you want. So that's very important to look at not just one single antivirals, but to actually look at the usage of multiple ones to actually prevent this possibility. And then usually once you characterize one antiviral, the subsequent step is to actually monitor the possibility of the virus evolving and becoming resistant as well. That's part usually of the series of work you need to do while you characterize an antiviral.
Very good. I have a question from the audience in terms of the other projects you highlighted in relation to the magnetic beads in terms of isolating RNA. It's asking, and you may know from your work with chemistry labs involved in the various projects on this, but what modifications are considered for the magnetic beads in order to make them more selective for viral RNA, do you know?
That's more the area of expertise of Dermot and Suzanne. So they have a whole chemistry behind it that they are optimizing at the moment. So they're still working on it and selecting the best way to actually modify the beads. So I cannot say much at the moment, but hopefully we'll be able to put the data together and give more.
Very good, very good. Just then as well, in terms of collaboration, you kind of did mention a little bit there, one of them, that the various work in terms of the antiviral project has led to potential other questions to answer in terms of collaborative research. Do you see a kind of a scope where the various efforts in COVID-19 research globally has encouraged more collaboration, or is it more competition between various different research groups or universities?
I think what we've seen in terms of, and I think we've all seen this from the news, in terms of vaccine research, there seems to be a lot of competition, but because it's maybe driven by the industry and they really want to get their product out there on the market very quickly. In terms of basic knowledge, molecular virology and antiviral drugs, I think maybe it's more collaborative, I would say, definitively. Like when I started to work with SARS-CoV-2, although it's a virus, it was a new virus for me. So I quickly reached out to my network of European colleagues who were actually already working with like SARS or MERS and so on. And they were very responsive and helping very quickly. In terms of access to reagents as well, it happened very quickly that people developed new cellular models or optimized cellular models by overexpressing typical protein that we need to actually, optimize viral replication. And again, like every time I contacted them, although I didn't know them, quickly they were responding very quickly and giving me all the tools basically to do my research and to develop new antivirals. So I think there was a lot of collaboration, and it was quite positive in my view.
It's certainly been a fascinating year in terms of science research, in that for the first time the globe is focused on one problem that is affecting everybody, and everybody's willing to facilitate progression in that area of research, where you probably would, I imagine, see still competition here and there, but it's a bit of a 50-50 from my experience as being in academic research in the past, where you'll have 50-50 in 2 labs competing to get to an end goal, or you'll have 2 labs collaborating to get there together. And it certainly has offered up a situation this year to allow potentially greater progress I would imagine. So I was curious to know your take on that. Thank you for that.
No, sorry.
No, go ahead. Sorry.
No, I think it was a brilliant opportunity as well to meet, even within UCD. Like it's so vague and big, and actually to meet different people and from different background and area. Yeah. Yeah,
and that's one of the focal points of this seminar series in itself, is we want to facilitate more of that, you know, because only good things can come from it on our minds. So hopefully that continues. We're kind of running slightly just over time. I just wanted to end with one quick question in terms of your own typical work in terms of viruses before COVID-19. Do you think that the work that's going on at the moment, either by yourself or others, is going to help facilitate or progress those areas of study that you're working with, say, for instance, in HIV? Do you see a lot of benefits outside of COVID-19 research that will stem from the
so far, to be honest, I see more. So we obviously I am not the only one in the lab. Yeah, so we were big teams, and everybody just pulled up their sleeves and started the work. And all of them had their own project on HIV. So what we had to do so far is really to park on the side our HIV cure project. So far it has slowed down what we were doing on other viruses. And I know we are not the only one. I've seen this as well with my collaborators in Europe, where they actually had to stop what they were doing and really focus on SARS-CoV-2. Now, I think what we're generating, all this information, we didn't know so much about the coronaviruses and how they hijack the cellular pathways and this host virus interaction and the impact at the individual levels as well, like inflammation and cytokine storms and so on. I think we're learning a lot from that. And I think it will prepare us certainly for the future. It's a pity, I have to say, that you had already a lot of information about the first SARS, and you had group working on it, but they pulled the plug on them, like they stopped the investments because they thought it was a finished story. And so we had animals models that were in development at the time that were actually put back into the freezers or the liquid nitrogen and so on. And we lost a lot of time. I think we could have done so much better if they had sustained these groups that were working on coronavirus before, and the same for other kind of basic research, fundamental research.
The lessons learned. So, I'd like to thank you, Virginia, for joining us today to speak about your various different research into COVID-19, and also thanks to all the attendees that joined us today and taking the time to do that and your questions. If anybody would like to continue the conversation or submit any further questions that we can get to Virginia, please engage with the COVID-19 Basecamp team that you would have registered to when you were signing up to today's event. And just to say that next week we'll be continuing our seminar series. We have another session that will be with Dr. Carla Kanan with Professor Niamh Moore-Cherry and Alma Clavin, and their research topic will be cities under lockdown, public health, urban vulnerabilities, and neighbourhood planning in Dublin. So thanks again to Virginia and to everybody for attending. Everyone have a good week.
Bye-bye.